David Kennedy. Now, you've seen his name pop up on a few occasions in these early pictures. Dave was actually another in the line of spectroscopy, exactly, chemical shift imaging types. Dave, you may not know, was actually the first to do multiple quantum chemical shift imaging. That's what his thesis was about. But like others, found that there were easier ways to make a living in NMR than doing spectroscopy. And in particular, through connections with Alan Nelson at MIT and others, developed an expertise in computer image analysis. And so during those early days was kind of the master of how we actually analyzed our data, how we generated those functional maps, the actual computational tools to do that, all were developed by David. David, of course, today is a professor of psychiatry at UMass, was the founder of the Center for Morphometric Analysis, kind of the precursor to the Free Surfer Lab that we may be more familiar with today, but quantitative morphometry, the brain was done at the Mass General well before Bruce Fischel and Anders Dale came on the scene, and it was Dave that developed the computational tools to do it. But in his, I think, amongst his very earliest experiments were to analyze the data from functional imaging experiments. So I thought it would be great to have him talk about the days of how they actually generated those beautiful early functional maps. David.
Thank you, Bruce. And so you already helped me with part of my introduction, is to say, fortunately, multiple quantum imaging was way too hard, and there were lower hanging fruit, as the previous speakers have all alluded to. You've also foreshadowed that while working on my PhD thesis with you, so I guess that makes me a great-great-grandson of Mansfield. I had been absconded with by the neurologists who got a little annoyed with the radiologists now that they had this beautiful imaging of the brain. You would take a patient with various neurological disorders, and the radiologist would say there's nothing wrong with it. Take a kid with autism, take an adult with schizophrenia. From the MRI point of view, those looked perfectly fine. And that annoyed Vern Cavanis, who walked across the river to find Alan Nelson and find a computer analysis lab and say, can't we do more about analyzing the information here? Can't we measure something? The brain is not right in these subjects. We need to do something in order to tease apart that information. So concurrent, while working with Bruce on multiple quantum, I was working on image analysis techniques. Now, in going backwards, 25 years, to tell a little bit of these stories about the first functional MRI experiments, it's quite surprising how limited our computational technologies were. There was not a workstation. You cannot just put a computer down, turn it on, have a mouse, have a image show up on that screen. All of that kind of had to be invented. And looking back, it's all relatively trivial to us today, but an awful lot of stuff had to happen to make even the simplest things happen. So while I'm giving some of this talk about the technology, Mike Vivi ran the imaging center and was clearly instrumental in making all of that work. Robert Weisskopf wrote an awful lot of the software that went into the generation of all these maps. Tim Davis generated some of the first image visualization tools that we had. Mark Cohen did some of the visualization tools. Again, a huge team of people were necessary to invent essentially everything. Now we have the free surfers and the FSLs and the SPMs. While SPM probably existed, it had no idea about an MRI scan. Is it live or is it Memorex is the quote that Jack started the 1996 OHBM conference in Boston with. And that, again, echoes something Bruce was mentioning, that while all of this driving, trying to understand how the brain working, was really dedicated and very focused to trying to preserve consciousness, to create a long recording and an ability to replay consciousness and long way going forward. So that, again, drove a lot of the technologies and the mission that we were all brought along with along this historical era. So armed with the tools that you've heard about from the previous speakers, we had the ability to measure blood volume. We had the knowledge, about where the visual system in the human is. And again, it was very nice that Arno foreshadowed that Jack targeted the visual system as the first objective. So the year now is about 1990 or so. Jack and I had the fortunate ability to go to the Dartmouth Summer Institute in Cognitive Neuroscience. Again, very early days. Not much was happening in imaging. There's some PET imaging, but we got to spend time with Peter Fox at that session and learned a lot about how PET was understanding the human visual system. And so really driven by the abilities of PET to take these types of measurements, that really set the stage for vision is the place to start, and taking these technologies of cerebral blood volume that you just heard about, can we do that experiment? Can we repeat the PET experiment using this MRI system? And back in those days, much of what MRI did was try to repeat all the things that PET could do. So if PET could do it, we could do it better, faster, and with less invasiveness than PET. So that drove a lot of the technologies in that era. So we knew well where the visual cortex was in general, but the visual cortex is a wandering beast, as most functional regions are in the brain. So while we know that the visual cortex lives in the banks of the calcarine fissure, where the calcarine fissure is in any given subject can be quite variable. So when it came time to do this experiment, the scanner was in Bay 3, I guess, of the Charlestown system. In order to focus our ability to detect the visual cortex, we used the surface coil for the brain, which then limited our field of view. So one of the biggest challenges, given our nice, comfortable subject, fitted with Peter Fox's own visual goggles, that were part of that experiment, of the original PET experiment, we had to initially find out where to put our imaging. Now again, nowadays we're all used to multi-slice, whole brain imaging. In those days, we had one slice. Yes, we could get 100 millisecond temporal resolution, but we could get that in one slice. In part, it was probably just due to memory issues. I think was the only thing that limited how many slices we could get. But heck, with 100 bits, you can get only so much. So one of the opportunities that, given my background with Vern Cavanis in the Center for Morphometrical Analysis, was to be helpful in trying to decide where we could put our one valuable slice. The first thought is, oh, we'll just put it, coronally or axially, just slice somewhere through the back of the brain and hope, to see that. But it was, I think, fortunate to us to again realize that the individual conformation of the calcarine fissure was a good place, to concentrate that slice on, so that for each of the subjects, we painstakingly, did an anatomic scan, studied where the calcarine fissure was, and prescribed, the slices, to go directly along on that particular orientation. So not unlike the types of pictures you've just seen, the subject, as we saw, comfortably fitted and stabilized with the goggles, undertook two of these boluses, once with the lights on and once with the lights off. The cerebral blood volume mapping was done in both those situations. And then a percent change map could be generated, where the difference between those two conditions could be observed. And so the first sort of task activation functional maps looked very much like the image on the left there, that looked at the difference between the lights on and lights off cerebral blood volume maps. And you could look at the brightness and darkness of that, but following, again, the lead of PET imaging, we developed the color scales to look at that in order to be able to set some sort of threshold that sort of detected, not quite in a same statistics that we do now, but a place where the difference was significantly different. In this case, 2 standard deviations change between those two different conditions. The slide on the bottom right just reminds us that we are looking at a slab of anatomy, and there is gray and white matter tissue in there. And again, we're in the neighborhood of where that calcarine fissure should be. But again, these are completely distinct acquisitions, and what's now trivial to combine those took a bit of work and registration and other elements to be able to accomplish that. Nevertheless, we were able to do that in the first 7 subjects, and this figure here sort of summarizes that distribution of visual activity that we saw. So it's 7 different subjects. From the sagittal views, you can see sort of the variability of sort of where that calcarine fissure was in those 7 different subjects. And in a standard space, in this case the Talarex space, again adopted from our PET friends and colleagues, we can see where that observation was and the extent of the visual activation as observed. So looking sort of in summary across the 7 subjects, we were to very clearly quantify a robust region of activation that was quite similar to the types of places that positron emission tomography was putting that observation. In the same time, we replicated the observation about exactly how variable anatomy is, even for something as simple as the Calcarean fissure, the primary visual cortex. That type of spatial variability will be an important aspect of much of the rest of the issues about how to do better mapping in the end, is to be able to understand and control for that spatial variability. We did it by observing in a particular location. Now there's much fancier techniques for dealing with that kind of stuff in free surfer, et cetera. So fine, that was great. We had a lovely story, but we need to sell that message. Jack wrote, with some helps of many colleagues, a lovely manuscript, and that manuscript was accepted, but we still needed to come up with a way to really make that hit home. This was a brand new technology. We were going to change the world by having this technology. We wanted the cover of Science, and we wanted everyone to know about this. So what do you do to get the cover of Science? When all we had was a percent change map and a oblique, half-gone slice of the head. So we did have one other thing. We had done an anatomic volumetric scan of that same subject on a separate occasion, or as part of that same study, but not as from the functional activation point of view. Now, because I just wrote this presentation recently, I didn't actually have some of the original data that was under that. So I'm going to use some sort of placeholder fill-in pictures to sort of show the concept of what we had to do. Again, you can do this today in 10 minutes. This took us months to deal with back in the day. So the concept was we wanted to show the experiment as in one catch-all picture of what was going on and what we observed. So the idea was to generate the human head, in this case a surface rendering of the volunteer's head. Again, a task that you can't just pull out Osirix and do. We did have access to an array processor from Sun Microsystems that had software that let us do this. So while it was doable and we didn't have to write that code, it was still effortful to make that happen. That view itself needed to also, though, be able to show us where we were actually observing in the brain. If we had the whole surface, you'd never be able to see the activation that we were interested in. And again, this is simulated now based on the types of things that we had to accomplish at the point in time. So in this case, that's just an axial cut into that. In our case, we had to figure out how to get the right obliqueness to get that cut into the head turned into such a way that would expose the exact slice that we observed. Fortunately, the MRI scanner knows the angling of the scans relative to each other, and we were able to recreate those kinds of cuts and those depth into the brain based on the data that was part of the acquisition. The next step is to merge our activation map, our percent signal change map, with our anatomic scan. Now, it would be embarrassing, well, not quite embarrassing, it would be not as exciting to show that surface coil-based image on that lovely 3D rendering. So we fortunately had the full image of that brain as well. So we needed to get our percent signal change map onto that represented on that anatomic scan in such a way that the rest of that brain was also visible. And again, sort of simulating those types of effects. But in the end, we could generate, I don't know if this thing works, but the types of images that showed that activation on the complete slice that came from that subject. Then last but not least, those two data sets needed to be fused. Again, technology for that was not cut and paste, was not existing. We have to do the viewing angle transform, an affine transform, to get that particular view turned into the particular angle and the viewing angle that was necessary for sort of showing that all together in the right perspective. Now, it needs to be reminded that it took a very long time to decide exactly what view of the brain to use, exactly how much to show the functional image, but how much to show of the human subject, to remind people that these are actual observations in living humans that are still walking around with no radiation, et cetera. So eventually, with lots of work and debate and consternation, the final image resulted and fortunately ended up on the cover. A number of other versions and touching up of this image happened. Particularly Mark Cohen worked very closely with the science cover folks to make this work right. Despite it not being quite very dramatic, there's skin toning on the skin to help make that look a little bit more natural. There are versions that you can see on the website of the Halloween version that had dripping blood and things like that, but those never made it into press. This, of course, was November 1st, so it was pretty close to Halloween, so that was kind of on our mind for that. So that really is a little bit of the background to that first image that became the picture seen around the world. In this case, we have documented evidence of it appearing in France and in the South Pole, and I'm sure there are many other occurrences, and that iconic figure is shown in many cases. And so again, I thank you and thank the opportunity to tell you a little bit about the making and the experiment that went into that particular experiment.